If you know what you're talking about, it doesn't matter as much how you say it. But.
Okay, dazzle us with information about MADS transcription.
Speaking of not knowing what you're talking about, let's dive into MADS transcription factors. Let's keep going. Okay, it's a family of transcription factors where the primary sequence, the similarity of the proteins is across a wide range of eukaryotes, and I saw stuff in like mice all the way to plants. It covered a wide range. And then the MAD actually comes from the original proteins they found, which is MCN1, AG, DEPA, and SIRT. So we have the MAD box. I had a hard time finding on the K box. I was looking, and it was like they would say they're not sure what it does, and it helps with the C terminal, but it wasn't really anything specific. So you can see the N terminal, which affects the DNA binding, and then the C terminal affects the dimerization. And that was something that I learned too. I don't know why I didn't understand the whole time, but the dimerization is the actual pairing of the two. Is it the CARGs? The DNA? Anyway, it shows it later on.
Oh, yeah. So what you just described, remember in the lecture, is essentially the DNA binding domain and the transactivation domain. So you see how they all the transcription factors have that similarity in terms of structure.
And that was one thing that I was glad, I was glad that I learned, because you see how like it binds on one side and then the DNA folds over, and then so the N terminal and the C terminal aid in that process of folding. And so again, that's something I learned that was good. SRF, there are just a lot of different functions for MADS. Actually, that's a picture of the SRF. It helps in early gene and muscle-specific gene transcription. Method 2D, it helps in muscle-specific transcription, so you have animal as well as plant functions. Yeast transcription. And you have just a few other, again, yeast transcription, Arabidopsis, Antirrhinum-Magus, so transcription in just a wide variety of organisms. Petunias, tomatoes, tobacco, maize. So we can see that the MAD complex affects just a wide variety of organisms.
So how many are there?
There were, I found there were like 170 something in Arabidopsis.
In just
So,
and
it's
when you showed the structure, what kind of structure does it have? Does it have a helix? Is it a beta sheet? Is it coil? What kind of structure is that there?
It looks like a coil with the I remember Nick telling us in the last lecture, it was like a coil with a bridge and then another coil.
Is that blue part a coil?
Yes.
Yes. It's like two coils and then a bridge between them.
Okay.
That's my understanding. And then DNA binding, there are specific sites that the MAD complex is bound to. And so in here, just some specific places. I thought what I would focus on more, because there are just so many ways that you can go with it, but focus on the floral organ development. There's a proposed ABC model, like we talked about in lecture. You have the AA and the AB and the BC model. And so it's proposed, but again, they're not really sure exactly how it works. You have the two dimers. Tetramers recognize the two different DNA sites, and then those are the CARG boxes that we'll see a picture of later. And they're brought close to proximity by DNA bending, so you have the strand, and it just folds over on top of itself on specific sites. And here we can see a picture of it. And again, they're not really exact, as you can see, they don't even know really what constitutes some of these. And so basically each part of it will affect the 4 different worlds. And if you mess up any part of each segment, then you can get like a sepal where you should have a petal, and so it messes up the whole flower. And anyway.
Is AP1 a MADS box transcription factor?
What is it? AP1? It should be, because
do you know what the name is of AP1?
What the name is? I know it is AP1.
A pedal up.
Oh.
So if you have a pedal and it says A, that means, you know, without. So that's how they identified it by mutation. Is you didn't get pedals, so it was called A pedal up.
Right. And so again, you can see the dimers, and that was something that I learned definitely. And it's
So those are heterodimers.
because they're not the same. Okay.
AG is A gammas.
And I looked up some different graphs on, but I didn't understand the process very well. But they have different graphs, and it would like have like a big C and a big T and a little A, and it would apparently it'd be like if they were comparing the two different types to see how much was expressed or the similarities in it. But I couldn't understand it well enough to.
The height of the letters is the percent conserved.
Okay. And so I looked through a bunch, but I didn't know enough to. So basically, what I've gone through is about all I could really glean from it. But
Okay, so you didn't do see any phylogenetic trees for MADS?
no, I looked up how many there were in Arabidopsis. It was 174, but I didn't know the whole tree of it.
Yeah, if they do phylogenetic analysis, it would be like the basic helix or the B-zips, in which you will find there are clusters that seem to function in particular processes more so than others.
And apparently, I mean, it has a wide range of functions as well. I mean, because like whenever we wrote it down, it was just talking about flowering, and then when I got to looking at everything else it did, there were a lot of different functions for it. So.
Yeah, it's a big family. It's a big family. So of course, the most well-known, or probably the most studied characterization of it is the flowering.
But they don't know exactly how it works, even though it's the most.
Given, when you look back at what we knew back in the 90s, it's like we know so much, but we still don't know anything. It's sort of like somebody's studying AIDS. We know so much about the virus, the protein, the code, how it's transferred, the genome's been sequenced, but they still don't know how to control it, really. So you know a lot, but you still don't know everything you need to know. But you learned something.
Yes, ma'am. Yeah, and again, I've never even heard of MADS or really ever studied transcription factors, so it's definitely a learning experience.
But you have a better understanding.
Yes, ma'am.